Alyssa Grodell, and my report is on the CACNA1C gene and how it is linked to schizophrenia. The purpose of this bioinformatics project is to determine the difference between essential and non-essential genes. An essential gene will have a slower rate of evolution than non-essential genes, since variation is unexpected in an essential gene. A molecular clock is the average rate at which a species' genome accumulates mutations. It is used to measure their evolutionary divergence. Gathering information through rate tests and comparing those results with other genes can help determine whether or not a gene is essential. Compared to other genes, CACNA1C has a slower rate of evolution, indicating that this gene is essential. I found the CACNA1C gene through the phenotype-genotype integrator search for my phenotype of interest, which is schizophrenia. Then I used the DB-SNP search program to identify the SNP within the gene that correlates with the phenotype. Gathering that information, I used a program called UGene to generate sequence alignments between 14 species, as well as different phylogenetic trees. UGene also provided data to create tree reconciliation and rate tests. These rate test results were then used to create a molecular clock that can show the average rate of evolution compared to other genes. Schizophrenia is a serious mental illness that affects how a person thinks, feels, and behaves. Schizophrenia affects approximately 24 million people, or 1 in 300 people worldwide. People with schizophrenia may seem like they have lost touch with reality, which can be distressing for them and their family and friends. The symptoms of schizophrenia can make it difficult to participate in usual everyday activities. Schizophrenia has no known cause and no effective treatment, even though it's quite common among the population. I'm very interested in what could cause this disease that can distort reality. The SNP, or single nucleotide polymorphism, in the CACNA1C gene is known as RS1006737. Genome-wide association studies have repeatedly identified this SNP, which is an intron 3 of the gene, to be strongly associated with schizophrenia and bipolar disorder. LitVar is a program that provides an accurate search of variants and related information from human-related biomedical literature. According to LitVar, this SNP is found between the precentral gyrus, inferior frontal gyrus, and the superior temporal gyrus, and it is a missense mutation from AA to AG or GG. Dr. Liu in PubMed states that the CACNA1C gene is defined as a risk gene for schizophrenia, and a large genome-wide association study of European ancestry provided by the Psychiatric Genomes Consortium. This gene codes for voltage-gated calcium channels, which are the primary mediators of depolarization-induced calcium entry into neurons. It contains 56 exons and is located on the short arm of chromosome 12, region 1, band 3, subband 33. There are a total of 1,530 associations, which indicates that it is very well studied. The gene contains 6,515 base pairs of linear mRNA. It is within the families of ion trans, CAC1FC superfamily, calcium channel IQ, and GPHH. This gene provides instructions for making one of several calcium channels. Calcium channels, which transport positively charged calcium ions into cells, play a key role in a cell's ability to generate electrical signals. The importance of the gene has been shown through knockout experience with mice. The mice who had their CACNA1C gene removed experienced a decrease in memory, sociability, and depression, as well as an increase in anxiety and stress. This gene tree was generated through UGene, and the data will be used to create rate tests and tree reconciliation and alignment. The tree alignment allows for further analysis and helps gather information about the sequence as a whole. Tree reconciliation displays opossum as the outgroup and has some differences from the species tree. This indicates that this gene did not evolve at the same rate that these species did. Since the tree reconciliation informed that opossum is the outgroup That species was used to create a rate test. The rate test concluded the average rate of evolution of CACNA1C. This information will aid in the formation of the molecular clock. The molecular clock graph displays the slope of evolution for this gene. Since it is quite flat, that indicates that there is a slow rate of evolution. In comparing my gene with the genes that my peers chose, as well as some housekeeping genes, my gene has a slower average rate of evolution. This indicates that my gene is essential. According to my findings, CACNA1C is an essential gene. Therefore, variation within the gene is unusual. Knockout experiments in mice displayed decreased mental function, but no deadly effects. Research shows that SMP RS1006737 is strongly associated with schizophrenia. Further study could result in advancements in the prevention and treatment of schizophrenia, which could change the lives of many people who suffer from this disease. Thank you.